Welcome to the spotlight presentation of Lexogen's NGS services arm. Lexogen NGS Services is a leading NGS service provider successfully supporting cancer drug discovery, with many of our clients focusing on precision or individualized therapies in cancer. My name is Eva-Maria Rösmann and I'm a product manager at Lexogen. Lexogen was founded in 2007 in Vienna, Austria, and has a subsidiary in the US. We are a company of RNA scientists that develop innovative solutions for measuring transcriptomic signatures. We develop and manufacture RNA-seq kits and distribute them worldwide. Our proprietary technologies are integrated in the NGS services facility to run complete RNA-seq experiments. We also act as NGS development partners and support the development of complete workflows for our customers to have them later fully implemented at their site. Our product portfolio spans the entire RNA-seq workflow, from sampling to data analysis, with various library prep solutions. I would like to highlight especially SLAMseq, used for high-throughput kinetic RNA sequencing, for which Lexogen holds the exclusive license for, and our in-house produced spike invariant controls, which are important for workflow validation and normalization. And today's focus will be on our NGS services in particular, and why we are a valuable partner for applications during cancer drug discovery and precision medicine. Compared to our product offering, our NGS services are not limited to RNA-seq only. With the involvement of our R&D department, we are able to tweak our proprietary technologies to meet our customers' high expectations and adapt the workflows to their needs. We also offer custom services and other state-of-the-art technologies outside of RNA-seq, which includes, for example, droplet-based single-cell sequencing using 10x, or multi-omic approaches, especially the combination of RNA and DNA-seq, or we also offer pure bioinformatics services. We have different Illumina sequencing platforms and also alternative sequences like the Aviti from Element Biosciences. So what makes us stand out? We use our innovative and cutting-edge technologies in combination with our wide NGS and RNA expertise, for example, in processing challenging samples like FFPE or blood samples, to customize our workflows to the market needs or the customer requirements. We thereby realize projects starting from small pilot experiments with just a handful of samples to large-scale projects with 10,000s of samples. And we do this by maintaining highest data protection standard as a US-based provider. And we always foresee a strong and sustainable partnership for long-term collaborations. And with that, we have established valuable partnerships with known pharma and biotech companies worldwide already. Our NGS services find application throughout the entire drug discovery workflow where NGS readout is beneficial. So we, for example, support target identification and validation with numerous applications, are able to support upscaling and high throughput screening during compound screenings, and are also experienced with pathway analysis or mode of action studies during hit to lead. Besides early drug discovery, we can also support preclinical and clinical stages, for example, with dose-dependent expression profiling, biomarker analysis, and expression monitoring. The following three showcases or examples showcase services customer projects on FFPE samples, blood-derived biofluids, and using an AI-driven approach I will present their achievements and highlight how we supported our clients with our tailored services and solutions. So successfully processing FFPE samples is one of our specialities, and Lexogen has also launched a dedicated FFPE product line for different RNA-seq approaches. And here I would like to walk you quickly through the scope of this customer project. This is the project of one of our long-term collaboration partners, who we supported with our NGS services during the development of their personalized cancer therapies. And it showcased the target identification and validation of neoantigens for cancer immunotherapy from FFPE samples. Neoantigens and neoepitopes are promising targets for cancer therapy, and their discovery and validation has built the foundation of individualized treatments of cancer patients. Combined DNA and RNA sequencing of patient-derived FFPE samples was used here to identify tumor sequences for a novel development of a cancer-preventing vaccine. Cancer mutations are foremost dedicated by DNA-seq. However, in this presented study here, it was shown that the integrative approach using RNA sequencing in addition to DNA-seq significantly improved the detection of neoantigen candidates and embodies a great potential for selection of neoantigen or neoepitope selection during personalized cancer immunotherapy. This was one of the reasons why our clients chose also the combined NGS approach. So what was the workflow of this project? With long-term projects like this, thorough consultation is key to grasp the objectives and the overall goal and align planning of sub-projects to it. Of course, we always keep reproducibility, standardization in focus, and this is really a main priority. And this is always considered during consultation and project planning. As a next step, the RNA and DNA were separately extracted from FFPE curls, and we generated libraries for whole transcriptome and whole exome sequencing. And in some experiments, we also used gene-specific panels. The libraries were then sequenced on both Illumina and Element platforms with comparable output. And then the data was transferred to our customers as they were doing the data analysis and interpretation at their side. And based on their findings, we would set up the next round of testing and the next round of experiment. And we are looking forward to some future breakthroughs and are excited to be part of this journey. The next showcase demonstrates our blood services. Blood or blood-derived biofluids are a common sample type in precision medicine and also heavily processed in our NGS facility. In this showcase here, biomarker identification was performed in blood-derived extracellular vesicles. Extracellular vesicles are small particles released from living cells into biofluids like blood or saliva. And they play an important role in the tumor microenvironment, and as they are shed from the tumor cells and often used for characterization or tumor diagnosis, prognosis, or also therapeutic purposes. And for this, they also are ideal for identifying cancer-associated biomarkers in a non-invasive manner in cancer immunotherapy, as they are loaded with various RNA molecules like mRNA, microRNA, long non-coding RNA, and also carry immunosuppressive molecules sometimes. In a series of service projects with a large pharma firm, we have analyzed numerous blood-derived extracellular vesicles and performed expression profiling using whole transcriptome sequencing and small RNA sequencing on them to decipher oncogenic signatures from tumor-derived extracellular vesicles and to target breast and hematologic cancer cells during cancer treatment. Over the years, we have processed various samples, blood-derived sample types, including whole blood, dried blood spots, plasma serum, and other blood-derived biospecimens like extracellular vesicles or PBMCs from humans and also selected animals. Patient blood is very precious and usually not available in high amounts. The RNA content is sometimes limited. The RNA quality can be impaired. And usually blood is a very individual sample type to work with. And the enrichment of the RNA of interest is therefore very important and not always very straightforward, especially for lowly concentrated particles such as extracellular vesicles. Special expertise is required in order to really process the sample type properly. In our service facility, we support various RNA-seq applications of blood-derived biofluids, including small RNA sequencing, whole transcriptome sequencing, 10x like single-cell RNA-seq of cell populations, for example, from PBMCs, but also high-definition single-cell RNA sequencing of enriched cells or subcellular RNA levels even. The last example shows how AI not only accelerates drug discovery, but also how RNA sequencing can be beneficial as an integrated process in this journey. This showcase demonstrates an AI-driven hit validation in combination with expression profiling for targeting cancer-associated microRNAs with small molecule inhibitors. MicroRNAs are recognized as key drivers in various cancer types and are therefore promising targets for developing anti-cancer therapeutics. Here in the study, molecular data was collected from multiple sources, such as cancer or toxicity assays, and phased into RiboStrike, which is a deep learning framework that can identify molecules capable of modulating the activity of microRNAs. The focus of the study was on microRNA 21, so miR-21, a known driver of metastasis formation in breast cancer. So RiboStrike here performed an in silico selection of 8 top small molecules out of a collection of 9 million. These were then validated through small RNA sequencing, gene expression profiling using Lexogen's sample bar-coded high throughput gene expression screening technology, Quantic Pool, and other reporter assays. And lastly, those results were also tested in xenographic mice, where the treatment with these small molecules actually showed a reduction in lung metastasis formation. So this study is a great example of how deep learning can revolutionize cancer drug discovery and furthermore demonstrates that RNA sequencing is a powerful readout for lead discovery. If you are planning a project that demands high throughput gene expression screening, ideally from cultured cells or blood-derived samples, I would like to draw your attention to our time-limited promotion for our high throughput services, which is also an ideal opportunity for evaluating Lexogen NGS services as a potential long-term partner for externalized high throughput, high quality NGS services. And with that, I would like to thank you for your attention and would like to invite you to follow us on our social media channels, LinkedIn, Twitter, Instagram. Yeah, thank you for your attention.